Hello, everyone. I am particularly excited for this session where our Simons NSBP scholars will be presenting their talks about the summer research that they did. I had the pleasure of meeting them in person, and I must say that I was blown away with how confident and how incredible these students are. And Dr. Alexander, he has always been a strong proponent for mentoring our students and giving our students a platform and opportunities. And I have witnessed this firsthand. And he was actually the visionary behind this program, where he wanted an opportunity for students to engage in research at the Simons Foundation, and these students had the opportunity to be in this fully funded program in New York City doing exciting cutting edge research. So with that, I would like to present to you the person who really had the vision for this program and made it from scratch into the incredible thing that it is today to create this amazing opportunity, and also the president of the National Society of Black Physicists, Dr. Stefan Alexander.
Thank you so much. One of our Farah, who's our student rep and leader of student council. So great. Thank you for those kind words. And I really can't take all the credit. I mean, but I appreciate that vote of confidence. I mean, yeah, so just a little background about this program. 2 years ago, kind of like when in the middle of the pandemic hit, David Spergel, myself, and Brian Keaton, who's the director of the Simons Observatory, we have known each other for quite some time. And one of the things that we always knew that had a lot of impact on our career choices and what we were going to do was basically access to real research opportunities, and especially outside of our home institution. So as all of us as undergraduates, we definitely knew that we had this, not new, but we had these research experiences as undergraduates at different places, and the connections that we made and the different perspective and the research skills that we picked up was very valuable in like really, congealing and giving us a window into a life of research. And propelled us into the careers that we were all in. And so this was a natural thing to do. The Simons Foundation provides some of the world-class opportunity researchers in computational astrophysics, cosmology, quantum computation, machine learning, and it's also a place where the students can pick up skills in coding, and so very pragmatic skills. That's transferable to different careers. And yeah, it was, and the other thing is that the Simons Foundation provided housing, full housing in the village in a nice part of the city. And I just knew, I know that the students had a wonderful experience, not only just academically, intellectually, in terms of research development, but personally, and they were able to make new friends and make connections with faculty and researchers at the various institutions in the greater New York area. And I had the pleasure of being able to welcome you all, them in New York City in person. It was virtual last year. And I do know this is something that David Spergel, the president of the Simons Foundation, that he really takes. This is one of his priorities, this program moving into the future, and to know that it has a full support of Jim Simons and Marilyn Simons, and also incredible staff, co-directors, Casey and Hakim, who was on the ground floor from day one, making it happen. And I'm just very proud that this is happening. And I think this is, this is very much in the spirit of NSBP and kind of what we're about. And welcome all these students into NSBP universe, because here you will find that basically this is very much part of what we've always been about. When I was a student, when many of us were students, we had those opportunities, and we were able to get some of these opportunities coming to the conference and meeting with other Black physicists and faculty. And we're just expanding this opportunity into the Simons Foundation land. Very grateful for their support. And on that note, I'm going to pass it over to Casey Wagner, one of the co-directors of the program.
Hello, everyone. So I think we're going to get right into our talks. If you are in the audience and you don't know about the program, after all of the scholars present their work, I'm going to talk briefly about the program and ways that you can either get involved or what it looks like to apply, and some of the things that we do in the program besides just the fantastic research that our scholars are going to present. So let's get to the talks. The first one is Teresa Ross. And so Teresa, if you want to screen share and do your thing.
I'm sorry you couldn't hear me. Just give me a quick second to set up. Okay. All right. Hold on. Screen share. Okay, I think we're running into the same problem I had earlier. Hopefully this doesn't. All right. Are you seeing the?
We're seeing the kind of notes version. We're not seeing the full presentation.
Oh, geez, not again. Okay, hold on. I apologize.
Would it make sense?
Is that better?
No, it looks the same.
All right. Okay, let's do it this way. All right, how does this look?
That's what we should be seeing.
Okay,
so,
cool.
Go ahead and start.
All right. Give me a moment. All right. So hello everyone. My name is Teresa Ross, and I am an Earth Science Master's student attending North Carolina Central University. This summer, I conducted research at the Flatiron Institute in New York as a part of the Simons National Society of Black Physicists Scholars Program under the mentorship of Dr. Maytree Bose from the School of Earth and Space Exploration at Arizona State University. The subject of this research is an element called phosphorus, and it's essential for various life forms on Earth. In my talk, I'm going to be giving you all a brief overview of why phosphorus is important and how elemental phosphorus is delivered to Earth. Before I start, I want to acknowledge Dr. Martin Suttle, who provided samples for this study. Change. Okay. Did it change? You guys see it change? Okay.
Yes.
So where does phosphorus come from? Phosphorus, like a lot of elements in the periodic table, originate through nucleosynthesis in the center of massive stars. Here you can see a figure showing the life cycle of a sun-like star on the left loop and a massive star on the right loop. A massive star will progress through different stages of evolution, starting with burning hydrogen, helium, calcium, neon, oxygen, and silicon until it begins to burn iron, which is not a viable fuel source for nuclear reactions. This begins a process of core collapse, essentially the death of the star. When the oxygen-rich star reaches the end of their life processes, they explode and eject freshly formed elements and molecules into the interstellar region. The production of phosphorus specifically involves oxygen and silicon burning, with oxygen burning occurring at temperatures at about 2 billion Kelvin at the center of the star. Various phosphorus-bearing molecules have been identified in the interstellar clouds, like phosphorus nitride and various oxyanions of phosphorus. During various stages of planetary formation and growth, these molecules can be accreted onto planets. Scientists argue that planets, and more specifically Earth-Moon formation, occurs in two stages. The first stage of planet formation involves collision of protoplanets and asteroids in the very early stages of our solar system's history. This chaotic environment often led to the destruction of larger planetary scale bodies, but would often lead to the creation of larger planetary embryos in the first 10 million years of our solar system's history. If the asteroid produced during this process were phosphorus rich, then that would bring the essential phosphorus to Earth. The second way phosphorus could have come to the Earth was through the impact of carbon-rich asteroids after the moon forming event. This is often called the late delivery of volatiles, and scientists argue that hydrous impactors brought in water and organics to Earth after it was fully formed. This will be the second way to bring phosphorus-rich materials to Earth. So now that we know where phosphorus is synthesized and how Earth may have received this element, you might ask the question, why should we care about phosphorus? So phosphorus and other phosphorus-derived compounds are incredibly important to the formation of life on Earth. The Earth itself has its own catch of phosphorus, but 98% of it's in the core. And we get a small amount, the other 2% comes from cosmic dust rain and from the atmosphere. Phosphorus is so essential that it can't be replaced by any other compound. All living creatures need it, and there's no replacement for it. In phosphorus's prebiotic form, it could have been an important role in the formation of our oceans and the life within it. As for humans, our DNA is literally being held together by phosphorus and supplies us energy through phospholipids. 85% of the phosphorus found in humans are in the bones and the teeth. For plants, it facilitates photosynthesis, and without it in our environment, it can cause crop failures and famine. So we talked about the delivery of phosphorus during Earth's formation, but what about modern day delivery? What do we know? So meteorites are portions of asteroids either from the main belt or the Kuiper belt that broke off from the parent body and made their way through the Earth's atmosphere to land on the planet. In the middle panel, you can see a picture of meteorites collected from the NSBP program. Meteorites and extraterrestrial dust particles can be easily identified in Antarctica because of their sharp color contrast between the ice and the pitch dark fusion crust that meteorites create as a result of entry through the atmosphere. Cosmic dust particles have been collected from Antarctic snow using a sheave or a magnetic separator. The right panel shows a picture of a cosmic dust particle by flying planes in Earth's stratosphere. So my study involved the cosmic dust collection from Antarctic ice and sediments, and the aim of this project was to constrain the amount of phosphorus in cosmic dust and in what form it's present. So a quick introduction on what a micrometeorite or a cosmic dust particle is. Micrometeorites are particles derived from either leftover solar system formation or separation of small particles from parent asteroids. They come in many shapes and sizes, usually between 50 and 3,000 microns. On this slide, you can see pictures of these particles. Each container has about 200 to 300 particles. Most of the micrometeorites are destroyed through atmospheric entry. When the dust does manage to make it to Earth, it's categorized into 3 groups: melted, partially melted, and unmelted. Melted micrometeorites enter the atmosphere and undergo chemical changes due to high temperatures, such as mass loss and alterations. These chemical changes can obscure the geological history of the parent body, making it difficult to track its origins. Unmelted micrometeorites are almost unchanged by the atmospheric influences and can give us a glimpse into its origins. Unlike meteorites in our collection that are predominantly stony iron, micrometeorites in our inventory are largely from carbon-rich meteorites. Earth accretes about 40,000 metric tons of cosmic dust a year. That's about 14,000 fire trucks being dropped on the planet a year by just particles. Meteorites are not the subject of this study because they accrete at a much smaller rate than micrometeorites, about 3,000 to 8,000 kilograms a year at size ranges between 8 grams and 1 kilogram. The dust fraction is predominantly in the small size range between 200 and 250 microns in size, as you can see by this figure by Levin Brownlee. To give you an estimate of the size scale here, that's about 100 and 200 microns, about the size of a human hair. So it's roughly the size of a single human hair, the diameter. Knowing these dust accretion rates, I wanted to understand how much phosphorus is being delivered to Earth through accretion of micrometeorites. Phosphorus could be present as organic coating on the minerals with micrometeorites or mineral grains. So how do we analyze something that's just so small? To measure chemical composition of micrometeorites and known phosphorus contents, an electron probe microanalyzer, or EMPA, will be utilized. In an ultra-high vacuum chamber of an EMPA instrument, the micrometeorite will be bombarded with electron beams through an objective lens at a voltage of 20 kiloelectron volts and at about 10 to 20 Na beam current. High-energy electrons bombard the sample surface. The inner shell of the various elements in the sample are ejected by the beam. To replace these electrons move from the outer shell, move in, and release a characteristic X-ray energy, which is the difference between the energy of the two levels. The characteristic energies are specific to the elements present in the sample and will therefore tell us chemical compositions of the micrometeorite. An example of some elements chosen are phosphorus, calcium, titanium, and magnesium. So far, we've collected phosphorus content measurements in 1 cosmic dust particle. So as you can see, here's a backscattered image of a mount containing 80 particles that we intend to probe for phosphorus. The electrons striking the particle will give off a signal that lets us observe the micrometeorite with more focus. Backscattered electrons are produced by the incident beam and are highly energetic. They travel deep through the sample, allowing us to receive a detailed image of the composition inside the particle. Heavier elements tend to be brighter because the backscattered electrons travel deeper within the sample, creating a larger interaction volume, which creates lower spatial resolution. On the right of the picture, you can see a single micrometeorite that is about 80 to 60 microns in size. You can see the brighter domains or regions that contain high Z materials, like iron and magnesium, while the darker materials could be carbon-rich. We'll be mapping these particles to look for phosphorus-rich minerals or domains. Then we will accurately measure phosphorus abundances particles using the wavelength dispersive mode on the EMPA. So this past summer and in the fall of 2020, I have been conducting research on cosmic dust particles through literature review and their compositions and possible origins. I was also looking for any information about the detection of phosphorus in cosmic dust particles, which I wasn't able to find, but I did find that evidence of phosphorus-bearing molecules in molecular clouds. So where are the next steps for this research project?
1 minute left.
Huh?
1 minute left.
Oh, geez, okay. So this topic is my ongoing master's thesis work at NCCU with Dr. Bose. I'll be traveling to Arizona State University in December to collect elemental data on micrometeorites. Once that's done, we hope to use this information to calculate the amount of phosphorus delivered to Earth currently by way of cosmic dust accretion. We also want to estimate the amount of phosphorus that could be delivered to proto-Earth in the form of micrometeorites during late delivery. I intend to continue the search for Earth's chemical origins through micrometeorites in a PhD program, which I'm currently in the market for. Do I have time for a summary, or should I just?
Sure.
Okay, so to summarize, phosphorus finds its origins in oxygen-rich stars through nucleosynthesis. 90% of Earth's phosphorus is found in the core. And the remaining 2 could possibly come through cosmic dust rain and asteroid belt bombardment. A good portion was delivered to Earth after it was fully formed after the moon forming event. This number is important to calculate because it holds importance to plant life, human DNA. We are running out of phosphorus, and we are running out of phosphorus in its current usable form. Without it in our ecosystem, it'll devastate life on this planet, and it's just not a good situation. So this makes the search for phosphorus in its elemental form extremely important. Yeah, I mean, a couple more slides, but it's all good.
All right, great. Thank you, Teresa. I'm just going to tell the audience now, if you have a question, you can put it in the Q&A and we'll answer those I think we're ready to move on to the next scholar, but if during this session, if a question comes up, please put it in the Q&A and we will ask it. Next up is Cadus Worku. So Cadus, are you ready to share your screen?
Yep. All right, how's that look, everyone?
It's great. Okay, there we go. We can see it now. So
okay, great.
We're ready. Go ahead.
Okay, so this past summer, as a part of the SNSB program, NSBP program, I got to work with Professor Jo Dunkley and her team on a cosmology project. And in general, we want to find this given cross correlation. And I'll get into all the component parts of that, but it's a cross correlation between these 2 effects that we observed from the CMB. And so to give a bit of background, the CMB is the background of everything, background of all other astronomical objects, and gives us a lot of great insights into both the early universe and the current universe. And the most recent full sky observations were thanks to Planck, and so we were using data from the Planck satellites. The data produced were a series of maps, and there were, I think, about 7 or 9 maps, and there are a bunch of different kinds of resulting maps that are produced based off of linear combinations of these base maps, different frequency bands, and we wanted to look at the 2 effects, as I mentioned earlier, and the first of them is the ISW effect, or the integrated Sachs-Wolf. So this relates to galaxy clusters, or more so the gravitational potential well that they sit in. So in general, the CMB is at the background of everything. When a CMB photon comes to us with a cluster, in between, It will sink into the gravitational well of the cluster. The photon will sink a given distance and it will gain energy. It'll travel through this well and then it will leave at the other side of the well. But generally, you're supposed to have lost a given amount of energy climbing out of that well, and it should be the same amounts that you initially gained when you fell into it. But with the integrated Sachs-Wolf effect, dark energy plays a role. Due to the expansion of the universe, specifically the expansion of this cluster, its gravitational well, it gets a bit shallower while the photon is in the well. And so it travels a smaller distance leaving the well. And so it gains energy in this process. The other effect that we looked at was the thermal Sonia-Zeltovich effect. And this similarly happens in galaxy clusters. So most of clusters are this intercluster medium, this plasma that has a lot of free electrons. So the CMB photons, once again, at the background of everything, they'll interact with this intercluster medium with these free electrons, and they'll get reverse Compton scattered. And it can either show up as a blue shift or red shift, but it's Doppler shifted to some degree. And in here, this expression, Y encodes all of the information about the electron distribution of the cluster, and G encodes the frequency dependence that's completely known. And so we expect these two effects to be correlated because they sit in the same physical location. And we want to look at that with Planck maps, as I mentioned. This is just an example of one. So the two maps that we used were called the SMICA and the Y maps. And we did a few things with the actual map. You can see a bit of a faint streak here, and that's actually the plane of our own galaxy, the Milky Way. That's been mostly moved, but it's still kind of there. And so we used a mask to cover it up. We did the same thing with our other mask, with our other map. We also used a mask to cover it up. In addition to getting rid of the plane of the Milky Way, we also used point source masks rather. They're really bright point sources, especially closer in galaxy clusters, that can contaminate our data in ways that we don't want. So you can see some blocked out portions all throughout. And so we did a few things with these maps. The first is making a power spectra, and power spectra in general show you variance as a function of scale. And so this actually has, each of these plots have the same amount of information as a full map previously shown. So what it does is L represents basically the scale that you're looking at. So very large L, closer to 2000, is actually really fine detail, while smaller L's closer to 0 to 50 are really, really large details. So the effects that we were interested in happened at smaller L's because they're within galaxy clusters, and they're the greatest, or the largest gravitationally bound objects in the universe. And so we looked at variance as a function of scale, and we were able to do a few things. One was reduce the noise with correlating the cross mission halves. So when you cross correlate, you're essentially looking at two of these maps, and you're saying, what do they have in common, and what do they have that's not in common? And what they have that's not in common is this independent noise factor that we can get rid of. So cross correlating mission halves as the same data, but the first half of the mission, second half of the mission, as opposed to cross-correlating the full mission with itself, gets rid of left independent noise. So we did that along with correcting for some of the other things that we were worried about, like the mask and the beams, with a great Python package to master. And so making these cross correlations, what we were looking for at the end of the day was this effect that these two effects and how they were correlated in galaxy clusters, the ISW and the TSC. So there were some great theory papers that laid out what to expect. So here, this red curve is what we ideally wanted, or that's what we sought after. And to give some scale, remember this is log scale. This purple curve up here would be the actual black body of the CMB. This would be closer to this plot right here. But logarithmic once again. And what we ended up finding was interesting. We initially found a negative cross-correlation, an anti-correlation, which was weird. We didn't expect that. And so we thought about what could it be? What are some of the possible sources of contamination, or if this is a true signal? And one of the things that we tried was blocking out different portions of the galaxy. I mentioned that we had this mass that blocked out the plane of the Milky Way, but you can block out larger and larger portions of it. And so we did that. And this gives us some insights, this large plot. One of those insights is that as we blocked out more of the galaxy, this plot got closer and closer to the zero line. So one of the possible sources of contamination, the cosmic infrared background, cannot be it, because that's ubiquitous everywhere. This is about the dust in given galaxies heating up and emitting in all directions, similar to the CMB of a CIB. We knew that if it was the CIB, then getting rid of some data would just increase the it would increase the uncertainty, but not necessarily the strength of the signal, and that's not what happened. So we don't think it's the CIB. What's more likely is that it's actually the galaxy itself, the plane of the galaxy. Even though we're masking out greater portions of it, that doesn't mean this is still a completely clean cut of it. And if we look at this previous image, we were seeking that red curve. These are different scales, Kelvin and microkelvin, but the actual theory curve here would be very, very close to the zero line. And so we're not there yet, but we were able to show that you can reduce this anti-correlation by increasing the galaxy mass, which gives us some insight into some next steps that we can take. One of those next steps would be, instead of using the map that we used, the SMECA map, we can use the commander map. And these are a whole lot of given details, but the commander map much better characterizes the different foreground sources. There's the CMB, and then there's a whole lot of stuff on top of the CMB. The SMECA map is relatively simple in that it takes the things that are close to the CMB black body spectra and eliminates the things that are not very close. But the commander much better characterizes all the different foreground sources. And thinking about the anti-correlation, the reason why the correlation was negative seems to be, to the best of our understanding, that the Y map is a different methodology than the other maps. And so it can treat a lot of these same foreground sources as negative. It can give them a negative value. So if we're looking at the same portion of the plane of the Milky Way that's contaminating our data, it might be negative in one, but positive in the other. And so although that can intrinsically be negative in anti-correlation, it's in fact just a correlation, but it's because of the methodology of the map making. So being more particular about the kinds of maps that we use and blocking out the closer in galaxy clusters that are quite bright will help with getting a better read on this cross correlation. Thank you.
Great, thanks, Cadus. That was fantastic. There is time for a question if anyone has one. You can put it in the chat, or sorry, in the Q&A. You can put it in the Q&A or the chat. In the meantime, our next speaker, maybe Aaron, do you want to try to get set up?
Yeah.
Okay.
All right.
I'm not seeing any questions, so Aaron, as soon as you're ready. Our next speaker is Aaron Kabidi, and so Aaron, go ahead and take it away.
All right, thanks, Casey. So over the summer, I worked with Dr. Sultan Hassan to constrain cosmological and astrophysical parameters using luminosity functions. So before we start about what we did, the background is sort of important. Cadus discussed a lot of the important background CMB in cosmology in his presentation. The basic objective of cosmology is to study the origin and fate of the universe. And the information we get from to study cosmology is mostly from the CMB data, and CMB is found 380,000 years after the Big Bang. And based on the CMB data, there has been a standard model of cosmology called the Lambda Cdm model that has been characterized. And it has, it basically theorizes a bunch of things. The first one is the Big Bang. And the second one is it talks of the universe as being large. And when you observe the universe at a large scale, it's isotropic and homogeneous. And it also theorizes things that we have not observed yet, but we have strong signals for, like dark matter and dark energy. And the standard model can be fully described with 6 parameters. It has multiple parameters, but it only requires 6 to be fully expressed. And we're going to try to constrain 2 of those parameters called the omega matter. It's the total matter density in the universe, including the baryonic matter and the dark matter. And the other parameter that we're going to try to constrain is sigma 8. It's the matter density fluctuation. So as we can see in this graph, the universe starts with a big bang, and then there is this cosmic microwave background that there's inflation, and then there's a dark age, and after the dark age there's a period of reionization. So we're going to study the period of reionization around the redshift of 6. So we're going to be using luminosity functions. Luminosity functions are a description of abundance of matter in a set. And we're going to be describing luminosity functions of galaxies. And they're perfectly described by Schechter in 1976. And it was a power function spectrum, as we can see in this graph. We're working with a redshift of 6. So the x-axis of the graph is the abundance, and the y-axis is the luminosity over volume. And we can see that for higher abundance, the redshifts seems to behave in a power spectrum behavior, which we'll see in a bit. And we're going to be using the Camel simulation to study this. The Camel simulation is a simulation that is designed to specifically for an input for machine learning algorithms to predict quantities that define the universe as functions of cosmological parameters. And we use machine learning to extract the information from the following simulations and make predictions about the observations. The Camel simulation as a background is also made of 2 different simulations, the TNG collaboration in the SIMBA. And the SIMBA is an in-body simulation, while the TNG has both in-body simulation and magnetohydrodynamic simulations. What makes the Camel simulation different is that they have the data sets and the simulations are specifically made for machine learning. So in the Camel simulation, there are 6 parameters. The two cosmological parameters that we're studying, the omega matter and sigma 8, and in addition to 4 other astrophysical parameters like stellar density. And in the Camel simulations, there is a data set called 1P, and 1P data set is basically changing 1 parameter at a time and seeing how the luminosity function changes. And to find the luminosity function, we can't really find it in the simulation. We had to calculate it using a relationship from Schechter, and we can find that from the star formation rates. And by varying the star formation rates and by varying each parameter, we tried to see how the luminosity function would change and which parameter change would make a change in those parameters. So when we varied each parameter at a time, we see that the luminosity function changes in omega matter, sigma 8, and the star formation rate ASN1, while the other parameters don't really have much change. So this was basically a test to see which parameters we could use to constrain the parameters. So what we did after that is we created a parameter estimation pipeline. So the Camel simulation is a heavy set of simulations that is very costly to handle, and it takes a lot of time. So computation is very expensive, and it takes a lot of time. Machine learning makes things easier because we can predict things, and we can predict things accurately. So the parameter estimation pipeline that we had was we had the omega matter, sigma 8, and the stellar feedback parameters, since those were the only 3 parameters that had visible change, that caused visible changes in the luminosity functions. And then we give the parameters into the Camel simulation, and that outputs a luminosity function. And we train this output using an emulator that we created. And then out of our emulator, we're gonna have a test data. And then we had an ensemble sampler, the MCMC, which does a great job. So it runs everything multiple times. We run it for 10,000 iterations, which took about 2 minutes. and which basically, if results are close enough to the real data, it's going to save them. If results are not close enough to the real data, it's going to discard them. So the observed luminosity functions, if they're real, they're going to be kept, and if they're not close, they're going to be discarded, as we see here. And this cycle iteration runs multiple times until we get a correlation between the parameters and luminosity functions. And what we discover based on our parameter estimation pipeline is that we were able to constrain the values within 2 sigma levels, but we're unable to constrain the very good with a very good accuracy. So the black point we see here is a random luminosity function, and then we're trying to recover it, and this is the random luminosity function recovery. I mean, we are able to recover the degeneracy between omega matter and sigma 8. A lot of research in the past, like the research done by David Spergel in 10 years ago, or almost 20 years ago now, shows that there is degeneracy between omega matter and sigma 8 for low redshifts. We're able to have that, but it doesn't work for 3 parameters. So what we did was we removed the other parameter, and then we only tried using omega matter and sigma 8, and we see that we are able to recover our parameter within 1 sigma level, which is really accurate. And it's a very important point to make. After that, we see that our error bars are not constrained enough, are not tight enough. This error could be caused due to dust, due to us not having enough luminosity functions, and also due to other factors. Then what we did was we included the metallicity factor for 2 parameters. We see that it's constrained very beautifully.
1 minute left.
We recovered it very beautifully, and then we tried using the metallicity factor for the 3 parameters. And again, we see that we're able to recover everything. We recover the luminosity function within 2 sigma levels. So to achieve even better constraints, our future work includes we adjust for dust. Dust is a very important factor in cosmology. We can see that the BICEP research, for instance, was well, kind of a failure since they didn't account for dust. And we can include luminosity functions from different redshifts so that we have better data and better fits to our models. Thank you. I can, if you have any questions.
Fantastic. Thank you, Aaron. Very nice. Again, I'll remind anyone in the audience, if you'd like to ask a question, you can put it in the Q&A. And in the meantime, maybe our next speaker is Latoya Anderson. And so Latoya, if you want to start sharing your screen.
Okay.
Okay,
Great.
so Latoya, when you're ready, get started.
Great, thank you. So hello, everyone. My name is Latoya, and I was obviously a Simons NSBP scholar this past summer. And I am also a Brooklyn College student studying physics and computational math. And today I'm going to talk about my summer project, which is the ASE-PySCF interface for density functional theory. So the way we start is we start thinking about the electronic structure. The way we study electronic structures of any sort of material is by using the Schrodinger wave equation. So simply put, the Schrodinger wave equation is the total energy, and that equals the kinetic energy plus the potential energy. So this is a time-dependent partial differential equation, which, like I said, describes electronic structures. And for very simple structures, such as electrons, photons, and the hydrogen atom, we can solve for it exactly for the wave function. However, when we get to more complicated systems, it becomes computationally impossible to solve exactly. So this is where density functional theory comes in. This is an approximation to the Schrodinger equation, meaning that we have to use an approximation method to solve for the Schrodinger equation. And the great thing about density functional theory, or which I will abbreviate from now on as DFT, we can use it to treat realistic systems. And what that essentially means is that we can actually apply these approximations to real life situations when it comes to studying materials. It's also the workhorse for computational material science. And what this means is it's this really nice balance between, again, applying this to realistic systems, but at the same time isn't so computationally intensive unless we were compared to solving this equation iteratively. And finally, it's great for optimizing crystal structures and describes crystal vibrations. So for some examples, we can use it to describe the atomic structure of helium, the crystal structure of sodium chloride, crystal vibrations of the metal organic framework 5, which is what you see on the bottom, as well as successfully model semiconductor materials. Now, you can, if you want to, write a program, a computer program, to approximate the Schrodinger equation using DFT, but you don't have to. There are plenty of open source software programs that allow us to do this, one of which is Quantum Espresso. So this is an open source software program that performs electronic structure calculations, and it also does nanoscale materials modeling. And like I said, it performs DFT calculations, and it's also widely used by material scientists. So material scientists, including solid state physicists, condensed matter physicists, as well as quantum physicists or quantum chemists. And this software program in and of itself actually does have a published paper that describes the capability or the parameters of Quantum Espresso. And so scientists have been able to use this to model systems and therefore also produce publishable work. However, challenging to use. So this system does require have software requirements that are more unfamiliar by scientists. And it also has an input file that's also in a language that's not familiar to many scientists. So this is an example of that input file. So what this is actually called is a PWSCF, or plane wave, it's a plane wave file. And this plane wave file basically consists of setting up your actual calculation or initial parameters to perform the calculation, as well as setting up the atomic structure itself. And so what we're using in this situation is the self-consistent field. So it's a plane wave self-consistent field input file. So as you can see, it is not written in a language that most are familiar with. So just hold that thought with this file. The next one is the atomic simulation environment. So this is a front-end open source environment that is also used to study material systems. And it is written in Python, which is a language most of us are pretty familiar with. And if you're not familiar with it, then it is a language that is fairly easy to pick up. It's also flexible enough to support third-party software backend systems. So what I mean by that is ASE is the software package that you directly work with. It has computational tools to study electronic systems. However, if you have a preferred way of calculating your material structure, you can import certain supported other software as essentially a black box, meaning that you don't have to fiddle too much with the third-party software, perform the calculation all within the ASE environment. It's a simple and consistent way to drive those electronic structure calculations, and is also used extensively in quantum chemistry, material science, solid state physics, and molecular dynamics. So to give you a comparison, remembering back to the previous input file that you would need for a quantum espresso, we have the AS, right? So this is many less lines compared to the previous file that you looked at. And what's great with ASE is that it pretty much has a library of atoms and crystal structures, as well as a library of different types of structures that you want to impose onto your onto the atoms or molecules that you are creating. So all you have to do is call it, which is pretty much what's happening at the line number. And from there, you can read in that third-party software, which is what I was sharing before. Espresso actually refers to quantum espresso. You set up your calculation, and then from here, you use the quantum espresso to create that, to generate that calculation. It then ends up in rocksalt.calc, and then from there, you can visualize that calculation however you need to. Now, you might be wondering, well, why use anything else if Quantum Espresso is a program that many scientists use? As I shared before, it's pretty difficult to get it started. So this is where PySCF comes in. So PySCF stands for the Python-based Simulations of Chemistry Framework. And what this is another Python-based open source package, and it is performed it is designed to perform quantum chemistry calculations. It's easy to install. It can be used on its own, so you can use it as a front-end framework, or you can, the goal is to be able to use PySCF within ASE, simulations environment, so that you don't have to then use Quantum Espresso. It's also capable of incorporating methods that are not widely available in other software. So that's another advantage to using PySCF compared to Quantum Espresso. And so that's where my project comes in. So over the summer at the Simons Foundation, I ran electronic structure calculations on the Simons Foundation's high performance computing cluster. I also ran calculations using Quantum Espresso, used ASE, or the atomic simulations environment, to perform those calculations. I also drove Quantum Espresso, or used ASE to drive Quantum Espresso to perform those calculations. Essentially, that program that you saw earlier, I used that on the high performance computing cluster to perform those calculations.
1 minute left.
I also used PySCF to model solid state and molecular structures. And then from all of that information, I'm now prepared to connect or create an interface between ASE and PySCF. And so this is where future work comes in, which is actually currently my current work. I am working to connect these two packages together to generate DFT approximation, develop education materials so that not only material science can use these, but graduate students and even undergraduate students who are interested in material science. And that way we can expand the PySCF user base to the wider scientific community. In other words, we want to add PySCF to this list of logos that you see here, which are the third-party software that's available within ASE. And so with that, I want to thank the NSBP, Simons Foundation, my mentors, Dr. Tim Berkelback and Dr. James, as well as Dr. Casey Wagner and the CCA staff for creating such a wonderful experience this past summer. It really solidified my desire to go to graduate school for physics, as well as to continue studying material science. So much.
Fantastic. Thank you, Latoya. A reminder to the audience, if you have questions on anything, please feel free to put them in the Q&A or the chat if you'd like. Our next speaker is Tana Bua from Panoma College. So Tana, are you ready?
I am. Thank you, Casey.
Can you go ahead and share your screen as you're ready?
Yeah, can you? Oh, never mind. let's go back. Okay, can you guys see my screen?
We can. It looks really nice. So go ahead and take it away.
All right, perfect. Yeah, as Casey just said, my name is Tana. I'm a junior at Pomona College. And over the summer, I was primarily working for the Simons Observatory. So to get started, this is the telescope that I was primarily working with, which is a part of the Simons array, which is like a set of ground-based telescopes designed to detect CMB, polarization anisotropies. And this is the diagram that Aaron just showed you, which essentially just shows the evolution of the universe. So the Simons Observatory is really just mostly interested in the light that came from this part of the universe. And the Simons array will be located at the Atacama Desert in Chile, which has like a high altitude, dry climate, and will allow us access of 80% of the sky. So a significant improvement from the AG telescope. So before we get started, I'll have to talk a little bit about the data collection techniques that the telescope is using. So an observational strategy that we have is the inclusion of active polarization modulation in the optical path of the telescope. And this is achieved through a continuously rotating half-wave plates, which are located at the upper part of the telescope. And so this is the data that we have. And as you can see in this graph, there are all of these peaks that should not be here. So my job was to investigate these peaks and figure out where they came from and how we could possibly get rid of them. And in order to do this, if you can see, our data follows an exponential function. So I created a curve fitting algorithm using a chi-squared distribution with 2 degrees of freedom. And this is a histogram of the PSD plotted against like the data points. And as you can see, this little line shows how the curve fits to the data. And the way it picked out the outliers would be using a p-value that is essentially the probability that a random number drawn from the chi-square distribution would be larger than what we would expect to get. And this is just the equation for the chi-square distribution. This is an example of one of the channels that we used. I wanted you to see the peaks in a bit more detail. So for this one, we had primarily 2 categories of lines that we were interested in. So we had Ptc lines, which are from the pulse tube cooler, and we had lines that we suspected were caused by the flux ramp. And then we also had half-wave plate lines but those were previously investigated. So before we get to that, I'll have to talk a little bit about the different detector populations that we had, because we looked at the data collected from all of them to see if we would be able to pick out a certain noise from all of these different detectors. So this is a graph showing the voltage bias and the transition and the different channels and how they transition. And this is the pod where we'd call like pod 3, which is the normal. And then this would be the transitioning. And then this pod is the superconducting pod. And these are the frequencies at which the half-wave plates are rotating. So we looked at the plates that were rotating at 2 Hz and at 1 Hz. And we also looked at fluid data for all of the 3 different detector populations. And then to get started, as I said, we divided the lines into 3 categories. So we had Ptc lines, and these were lines generated by the Ptc cooler, which provides a cooling system for equivalent power for our system down to 4 kelvins. and it operates by oscillating helium at 1.4 hertz. So so the lines occur at increments of 1.4 hertz, ranging from 1 hertz to 40 hertz. And this is an example of how the curve fitting function worked. It essentially just looked at the different channels and picked out the appropriate and the peaks that we were looking for. So this is an example of a peak at 1.4 hertz. This is a plot of the frequency against the PSD. So here we have two graphs showing a detector channel, and we also had graphs for the resonators. And for these specific lines, we only got phase for the detectors, but not the resonators, which indicated that the detectors were the only one picking up these lines, which was kind of a clue as to what we were looking for. And then this is the range that I was talking about from 1 kilohertz to 40 kilohertz. So these are all the peaks that our algorithm picked out for the Ptc lines. And all the lines, as I said, are multiples of 1.4 hertz. So from here, I also plotted a histogram of the different amplitudes that we got to try to figure out the spread and to see if all the lines that we were getting had essentially is the same distribution of the amplitude. So this is a plot of the superconducting transitioning normal detectors, and we also had a plot for the resonators. And as you can see, there are no resonators because there was no pickup in any of them. And we got stronger pickup in certain detectors than others. And then from there to try to figure out where these lines are coming from, because at the beginning, we didn't really have a clue. We positioned a magnetometer around the remote motor and the Ptc motor. So this cryomag logo is for the pulse tube cooler, and this black thing is a magnetometer placed around its motor. And then from here, we place it at different points throughout the telescope to try to figure out where the lines were originating from and whether or not they were magnetic. So here is on the compressor, the motor, and then this is the compressor. And then this is above the window, so above the halfway plates. And then we got data from the magnetometer where we tried to see if the data that we collected from the magnetometer aligned with the peaks that our algorithm picked out. And I have an isolated graph of the data from the remote motor, which perfectly aligns with the peaks that we got. So we came to the conclusion that the peaks were indeed magnetic field lines from the remote motor. And then we also had lines that may be caused by the flux ramp, which is part of the modulation, the squared multiplexing modulation technique that we use. And we had we picked them out at these frequencies. So this is the same plot, but for flux ramp lines. So we had fizz in both like the detectors and the resonators. And from here, we also plotted, this is also another line which it shows the same thing. And then I plotted the histograms as well to see the amplitude spread. And from here, you could see that they don't really have the same spread. So they were from the same source. And then to figure out whether indeed they were
1 minute left.
Lines, thanks. To figure out whether they were flux ramp lines. So we had we changed the frequency at which the flux ramp of the flux ramp. So it was initially operating at 4 kilohertz. And then so the orange part would be the initial frequency of the flux ramp. And after altering it by a little bit to see if there would be a shift in the lines that we saw, we did indeed see the shifts in the lines. The blue part shows the data after the shift. So yeah. So from here, the plan is to investigate where the flux frames shift to and why they shift, and also investigate the lines from the remote motor that do not show up in the detectors. We also have to figure out the dimensions of the peak so we can incorporate them in simulations that for the data that we expect to collect, and to see to what extent though they will affect our data analysis. And I would like to especially thank Jake Spizek, who's an undergrad at a grad student at UCSD, who was an incredible mentor, Brian Keating and the UCSD SET testing team, and then Casey for being a wonderful program coordinator, and the NSBP and the Simons Foundation. Thank you.
Thanks, Tana. Very nice. So I think it's time we need to move on to our next speaker, which is Mr. Carlton Smith from the University of Florida, as you can probably tell. So Carlton, you want to share your screen?
Let me unmute myself first and share my screen. Okay. So let me just put this in presentation mode. All right. Thank you, everybody. Thank you, Casey. Thank you to my fellow panelists. Today, I'm going to present to you my work at the Flatiron Institute in the Center of Computational Neuroscience on a few of the theories of neural plasticity and learning as mentored by Dr. David Lipschitz and Dr. Dmitry Choklowski. So the first thing I'd like to do is I'd like to draw a comparison of artificial neurons that compose a neural network and biological neurons that compose an actual like neural system in a living organism. And so the first comparison I'd like to make is that neurons in the biological setting have what are termed dendrites, which receive input in the form of electrical signals. They have synapses that can strengthen or weaken, and they have an axon which delivers an output. And so we'd first like to consider in a single neuron layer network that that much like a biological neuron, input is fed into a node, and the strength of the influence of that input is modulated by a weight, and an output is typically delivered out, a Y, if you will. And so a bit of notation overview is that we represent either the weight vector or the weight matrix with W or capital W input vectors, X, data matrix, a capital X, output a scalar Y, or if we have a multi-neuron layer network, a vector Y. And we normally say that the dynamics of our weight vector for single neuron layer networks or weight matrix is dependent on the output Y or Y vector. And so the first thing I'd like to do, for those of you who aren't familiar with how neural networks work, or in general how machine learning work, is that we typically deal with the machine or individual trying to complete some task, and the measure of how well such machine or person completes a task can be quantized by what's called an error function. And typically machines would like to minimize the error function over multiple iterations. And they do this using a method called gradient descent, where we will typically take our cost function, consider our parameters. Here we refer to them collectively as theta, and take the gradient with respect to our parameters and move in the opposite direction of our gradient. So to summarize all of that, what we essentially do is we look at all of the parameters that could possibly affect how well our whether our cost function increases largely or if it decreases largely. And we alter our parameters such that the cost function, which measures how well we're complete in our task, decreases. And we can affect how quickly this how quickly we move along this gradient with a parameter called the learning rate. And so the field of computational neuroscience originally started in 1949 with an individual by the name Donald Hebb, essentially stating that inside of the brain, weights between neurons are altered such that if you have a presynaptic neuron and a postsynaptic neuron, the neurons will alter their synapses such that neurons that essentially fire together wire together. And as we go further along in the presentation, I'd like to list a couple of biological constraints that we use to determine if an algorithm that we believe the brain is computing is biologically plausible. And so the first couple of constraints are that we generally like to work with online algorithms, which are opposed to offline algorithms. Online algorithms take in a stream of information, so one input point at a time, as composed to an offline algorithm that will normally utilize the whole of your data set. We'd also like to consider local algorithms such that if there is an update to a weight, or in the case of, or in the biological setting, a synaptic strength, that update is only dependent or only is informed by the weights of neurons within its locale. We also would like to typically work with unsupervised learning algorithms, because typically the brain doesn't necessarily receive input that's neatly labeled into categories, and so that's largely the reason why we work with unsupervised algorithms. And so the first algorithm we're going to go over is what's called OHA's algorithm, or some variation of principal component analysis. And so the general goal of principal component analysis is to explain data in dimensions that best explain the variance that we see in said data. And so we will, in the following slides, consider the following cost function, and our traversal through the air surface will be determined by taking the derivative of this cost function. And so if you actually see on the slides here, you'll see how we can actually determine how our weights change considering this cost function in the online setting, where we have a stream of information, and the offline setting in which we consider a whole batch of information. And so just as I guess clarification for what kind of dimensionality reduction we're doing, we just took two Gaussian distributions, one along the x-axis and one along the y-axis. And essentially what we like to have our algorithm do is simply point out that most of the variation in our data is along the diagonal we see here and the perpendicular diagonal. And so the first algorithm we work with, well, the first setting we work with is offline OHA's algorithm. And so offline OHA's algorithm takes in the entirety of our data matrix generated from here and computes the gradient for each iteration. And as you can see, big
1 minute left.
as you can see, we have a situation where we have a large eta and a small eta, and you largely see less stochasticity. And we have online OHA's algorithm, which exhibits a large stochasticity in term when we have a larger learning rate and a smaller and a less stochasticity with a smaller learning rate. And I'm going to briefly go over the case where we have multi-neuron layer networks. And this is largely shown with similarity matching. And you can see that in this case, actually you could pair it here, that you have multiple means of computing. You have multiple means of computing this dimensionality reduction. And I will include here with any further research.
Okay.
Yeah.
Is that it?
Yes.
Okay, great. Thanks, Carlton. Like clearly you had a ton of work, even more than you had time to show. It was really fantastic. Thank you.
Yeah.
Okay, so our last speaker of this section is Bridget Gifford. And so Bridget, it looks like your audio is connected. Do you want to share your screen? And okay, great. We can see it. So whenever you are ready, you can start.
Alrighty. Hello and good afternoon. My name is Bridget Gifford from the University of Florida, and today I will be presenting predicting SARS-CoV-2 viral load from gene expression data using machine learning. All right, let's begin with some background. The initial study consisted of over 3,000 Marine recruits, over 1,000 of whom tested positive for COVID-19 at some point during the study. The goal was to learn more about transmission of the coronavirus among young adults. In fact, by studying Marine recruits in their close living environments, we were able to investigate how COVID transmission might occur on college campuses as well. So how do we do this? Molecular data and viral load, or how much virus is in an individual system, was collected for everyone involved. Additionally, gene expression was measured in intervals throughout the study, whether it be immediately before or after testing positive. With that background in mind, what are the key questions we are trying to answer in our research? First, can we predict based on the gene expression data collected whether someone has COVID or not? Second, can we predict viral load from gene expression data, in this case, Rna-Seq data? And finally, based on this, can we determine which genes are most predictive? As I mentioned in the previous slide, we measure gene expression by looking at Rna-Seq data. Well, that begs the question, why do we use Rna-Seq data? Some of you may be familiar with the central dogma of genetics. In simple terms, it states that DNA is transcribed into RNA, which is then translated into proteins. These proteins, of course, are responsible for keeping our bodies alive. Because DNA codes for our genes in the same proportion, it makes it hard to look for COVID-specific genes. Think of it as looking for a needle in a very big haystack. Instead, we look at RNA. Unlike DNA, RNA translates more of a certain gene based off immediate need. In other words, more of certain genes are expressed at a certain time. This makes it easier to find COVID-specific genes. All right, so now that we understand what it is we are looking for, how do we actually go about making a prediction model? The answer is machine learning. Machine learning algorithms build a model based on sample data called a training set. This is so it can improve and make predictions without being given the real answers. Then after the algorithm is done training, the test set is used to test how well our model actually performs. The only problem is that with large multivariate data sets, it's often hard to summarize or visualize the data in a chart or a plot. That is why PCA is used to reduce the dimensions of the data by finding a new, smaller set of variables that retain most of the training set's information. Within machine learning, of course, there are two main subsets, supervised and unsupervised. Both of my models use supervised learning, which is just that we have some prior knowledge of what the output values of our samples should be. The first model is a classification example of supervised learning using binary data. It predicts either COVID detected or not detected. My second model is a regression example of supervised learning using continuous data. It predicts the amount of someone's viral load based on gene expression. As I said before, my first model is supervised classification model that predicts whether COVID-19 is detected or not. What you're looking at is called an AUC ROC curve. AUC, or area under the curve, is the capability of a classifier to distinguish between classes. The higher the number is to 1, the more capable the model is. In this case, I plotted both the AUC ROC curve from the PCA data set, which has dimension reduction, and from the non-PCA data set that doesn't have any reduction. The two plots show very little difference, with the non-PCA curve having an AUC of 0.935 and the PCA curve having an AUC of 0.945. This is very good. All it means is that reducing the dimensions of the data had very little effect on the model's efficacy. It also means that the model can effectively distinguish between the positive class where the virus is detected and the negative class where the virus is not detected. My next model in my research aimed to predict the viral load based on gene expression. In order to study viral load, we look at the CT values, or the cycle threshold. This indicates how much virus an infected person harbors. To compute the CT values, we look at the number of PCR cycles that are necessary to spot the virus. You might be familiar with PCR tests, or the method that's often used to test to see if you were COVID positive or not. One of the most important things to note is that CT values are inversely proportional to viral load. The higher the CT values, the more PCR cycles it takes to find the virus, and the lower the actual viral load. So the lower the CT, the higher the viral load. Finally, the reason why studying viral load matters is because a higher viral load in an individual means they're more infectious. So studying CT values can help epidemiologists track outbreaks or identify people of high risk for severe disease and death. So now that we've discussed what CT values are and the importance of viral load, we look now to the results of the viral load prediction model. LASSO is a type of linear regression that utilizes regularization methods. All this means is that the model automates feature selection and elimination, leaving only the most variable and meaningful data. Now let's look at the plot results. Here I have made a plot to illustrate the prediction error of the LASSO model. The identity line that you see here is what the plot would look like if the model's prediction correctly aligned with the actual CT values determined. The best fit line is how well our model fits to the identity line. As you can see, there is a positive correlation. The predicted viral load follows the general trend of the actual viral load. It predicts relatively low load when it actually is relatively low, and vice versa. However, it's not a perfect model, which is to be expected. Predicting viral load from gene expression alone is a difficult problem with many factors. Still, the model shows that there is significant relevance to studying how involved gene expression is with viral load. In addition to the plot of the CT value prediction error, I also went ahead and made a plot for the CT values residuals. This essentially shows the identity line from the last plot, except this time is on the x equals 0 line. Above the line, we see how much the training set and testing set overshoots the actual CT values. And below the line, we see how much the training and testing set undershoots the actual CT values. As you can see, the points are pretty much evenly distributed with no specific pattern. We can conclude that there is no bias in either direction. After all, we wouldn't want a model that consistently over or undershoots. Finally, we should note that our model still has some overfitting. While the correlation coefficient of the test set is about 3.302, the training set's R-squared value is about 0.577. While this is not overly significant, it should be taken into consideration when moving forward with the research. All right, finally, the last question we aimed to answer was, which genes are actually most predictive of viral load? From the lasso regressor, we found that 48 genes had non-zero coefficients. Among them, 2 genes stood out in particular, LY6E and IFI44L. Both of these genes were linked to COVID-19 disease processes in previous studies. IFI44L codes for an interferon protein that acts to inhibit viral replication. LY6E, on the other hand, codes for lymphocyte antigens. Lymphocytes are just immune cells made in bone marrow, and it acts to restrict entry of the SARS-CoV-2 into the cell. So looking at the plots, we see that when CT values are high, gene expression is low. This makes sense. A high CT value means a low viral load. It would make sense that the less virus one has in their system, the lower their gene expression is to fight the infection. Now for the conclusions. We were successfully able to create 2 models based on gene expression alone. The first was a classification model that predicts COVID-19 positivity or negativity. The second was a regression model that predicts the amount of viral load. From the lasso regression model, we were able to isolate 48 genes that show the highest feature significance. 2 of the chosen genes were LY6E and IFI44L that were previously associated with COVID-19 studies. Finally, we can conclude that studies in viral load in COVID-19 patients should be continued. And before I go, I want to give a huge thank you to my wonderful mentors, Dr. Rachel Stilfon and Dr. Natalie Sauerwald, for giving me lots of support and teaching me so much this summer. And of course, I want to thank the Simons NSBP Scholars Program for giving me this wonderful opportunity. And thank you so much for your time.
Great, thank you, Bridget. So that's the last of our scholars to speak today. And I think now we have just a few minutes. I plan to give an overview of the program for anybody that's in the audience and is thinking about applying next year. I see that there's a question in the chat, in the Q&A from Winston to Teresa. So maybe I can find a way for them to connect offline, because I want to make sure that we can get over. We only have 3 minutes. So I'm going to talk real quick about just give an overview of the program for people that don't know about it other than the research you just saw. This is going to be fast. If you want to know more, there's a website listed there, Simons Foundation / Simons NSBP Summer Program -NSP. -SNSP. You can also email me. I'm Casey Wagner. You can email me casey.reed.wagner@gmail.com. This is a partnership between the Simons Observatory, NSBP, and all of the different centers of the Flatiron Institute. And you heard about a lot of work in these different centers. In addition to the research projects that you just talked about, every week during the program, we have a talk from a different professional Black physicist. And here are just 2 of the speakers that we saw talk about their work this past year, Professor Carol Scarlett and Dr. Delilah Gates. We also have weekly meetings where we talk about various career advice. And so these are 2 of our speakers from this year that we talked to, Dr. Sama Ahmed, who is going to become a professor of neuroscience at University of Washington in Seattle, and Dr. Matthew Debasai, who is actually an industry scientist working for Intel. We also do a number of other things as part of the program. This year, we actually took a tour of the CMS experiment at CERN, which is the Large Hadron Collider. So they walked us through the experiment. They had a GoPro and showed us. It was really cool. We had a visit to Princeton University, and this is a picture of our scholars that visited. This is actually Einstein's desk. And so they got to sit at it and take notes and make the desk happier than it's ever been before. We also did an outing where we went to the Harlem Gallery of Science, and our scholars were actually the kind of role models for younger scientists that are participating in events at the Harlem Gallery for Science. If you're out there and you're interested, please, please, please do apply. If you're an undergraduate member of NSBP, please apply. No matter what, we want your applications. I've listed here the things that you'll need to apply. And also on this website, within the next week, the application will be live, and the first round is going to be due by January 1st. But please do apply. If you know anybody that'd be interested in applying, forward this to them. You can also have them email me if there are any questions. And if you're a professional physicist and you're an NSBP member and you want to get involved, there's also opportunities for you. And please, again, email me.